Theoretical physics is a branch of physics that deals with the development of mathematical models and frameworks to explain and predict the behavior of the physical universe. Theoretical physicists use a variety of mathematical and conceptual tools to construct models and hypotheses that describe the fundamental laws of nature and the behavior of matter and energy at a wide range of scales, from the subatomic to the cosmological. Theoretical physicists often work in collaboration with experimental physicists, who perform experiments to test and validate their theories. Together, theoretical and experimental physicists work to advance our understanding of the fundamental laws of nature and to explore the mysteries of the universe. Without our understanding of the underlying mechanism of the nature, all kinds of today technology would be impossible. For examples, communication networking, global positioning system, GPS, high-performance computer and magnetic resonance imaging. The Institute for Fundamental Study, or IF in short, was officially inaugurated on March 14, 2011 by Naresuan University Council as a research center unit. From the first day to the present, IF members are working on the cutting-edge areas of research in theoretical physics to expand the boundaries of our understanding of the fundamental laws of nature, including cosmology, gravity, string theory, quantum information science and mathematical physics. We would like to emphasize that frontier research in theoretical physics is essential for advancing our understanding of the universe and developing new technologies that would benefit not only Thailand but the world society.
Okay. Good afternoon, everyone who actually watching online or actually in this room. I'm Steven Yukong. I'm going to be the host for this Colloquium talk. And so this is the first talk for this new year, 2025, okay, which is actually number 33. All right. So today we have guest speaker, which is actually a colleague of mine, Associate Professor Teeraphan Laomettachit from School of Bioresources and Technology, KMUTT University of Technology, Thonburi. So today we're going to talk about modeling cell cycle regulation and cancer drug resistance using Boolean approaches. Well, please welcome speaker. Are you ready?
Okay, thank you, thank you, Ajahn Lin, for inviting me to share my research today. So good afternoon, everyone again. My name is Teeraphan Laomettachit. I'm from Bioinformatics and System Biology program. And I am also affiliated with Theoretical and Computational Physics group at KMUTT. And today I'm going to present my research on the topic of modeling cell cycle regulation and cancer drug resistance using Boolean approaches. So let me begin by talking about the motivation behind my research, which tries to understand how living cells perceive signal from the environment and process this information and respond appropriately to the incoming signals. So in this way, we can think of each cell in our body as a computational unit. So in this case, each cell sorry, I have this coughing sickness for a couple of weeks. Hope it's not too annoying. Okay. Okay, so we can think of each cell in our body as a computational unit. So the cell monitors the signal from the environment and use it as inputs, and then do some kind of calculation inside the cell and activate or trigger appropriate responses in terms of cellular functions that that respond to the signal. Okay, so for example, there are molecules around the cells that can be detected as the signals, such as nutrient molecules, growth factor molecules, or stressor molecules. And cells respond to this presence of molecules by using different pathways inside the cells to process the signals, and then respond by inducing some cellular functions, such as the promotion of cell growth, the increase in metabolisms, or the inducing of cellular movement, for example. So if we look more closely into the cells to see the components that function as the information processors, then we see that there are a group of proteins, the receptor proteins and the signaling proteins. So on the surface of the cells, there are receptor proteins, which is this proteins. These proteins, they have some part that stick into the outside of the cell, and they monitor specific molecules in the environment. When they detect these molecules, another part of these molecules that is inside the cell can relay the information into another group of proteins, which we call the signaling protein, like this one. So basically, these are different types of molecules. Mostly they are proteins that interact to one another, regulate one another. And usually molecular biologists represent this network as by using the nodes to represent the components, such as proteins. And then the regulation between these components are represented by arrows. Okay, so for example, we have a protein called RAS that can activate another protein called RAF. And you see that there's a signaling cascade that go from RAF to another protein called MEK and ERK. There is also another type of regulation, which is the inhibition, represented by this kind of symbol. So in this case, another protein called P10 can inhibit the activity of PI3K. Okay, so the cells use this kind of network of proteins to receive the information from the environment, process this information, and then trigger appropriate responses, such as cell growth, proliferation, or migration of the cells. So in my research, I'm interested in how we translate this kind of information, the interactions between signaling proteins, into a mathematical model, which is a mathematical representation of the biological system. And once we develop the model that can capture the behavior or the details of the biological systems, then we can use this model to simulate the behavior of these cells and also try to understand the function or the mechanisms of this interaction that carry out cellular function. And in addition, we also want to use the model to predict something new. For example, we can predict the intervention of this network to get some therapeutic benefits when we, let's say, consider about the diseases that happen from the misregulation of this interaction. Okay, so let me give you example. So these are a tiny part of protein network that regulate the cell cycle of budding yeast. So the cell cycle is a process that the cells grow and divide, so making more cells. And there are more than 100 proteins involved in this process. So in this picture, I just show you 3 of them, KMUTT protein, SIG1, and SY5. And based on experiments, scientists can identify the type of the regulation or the interaction among these components. For example, we know that KMUTT inhibits the activity of SY5. SY5 activates the activity of SIG1. And these 2 proteins have a mutual inhibition relationship between each other. When we understand the interaction between them, we can also measure the activities of these proteins. The activities are the levels of this protein in the laboratory as well. So, for example, this one, the scientists measure the levels of these 3 proteins using a molecular biology technique, and then we can quantify the levels of this protein into this visualization. And these interactions are dynamic in time. Okay, so you can see a time series profiles of this protein. So you see that SIG1, the level of SIG1 goes up first and then go down, and then that followed by the increase in the level of KMUTT, like this. So when we look at the data like this, the straightforward way to represent the system as a mathematical model would be using a set of ordinary differential equations, right, to describe the rate of change of each of these components. Okay, so and actually we have a detailed model for the budding yeast cell cycle. We use differential equations to represent the rate of change of each component. And this change is a function of the other regulators protein. Right? For example, if you would like to keep track of the change in the level of KMUTT, that would depend on the activity of SIG1, right? And also, if you want to describe the rate of change of SIG1 over time, that would depend on the activity of SY5 and KMUTT. So we can write down the equations to describe the behavior of these components and do the simulation, just solve the differential equations. And the result would be something like this. And one of the advantages of using ODEs is that the simulation results can be directly compared to the experiments. For example, here we see that the simulation of SIG1 from this equation have similar behavior to what we observe from the experiment, right? SIG1 go up and then go down, and then that is followed by the increase in the level of KMUTT. Okay, so that's very nice. One limitation of using a set of differential equations is that we have a lot of parameters, kinetic parameters that we have to measure from the experiments or have to be estimated. And sometimes it's quite difficult to obtain the reliable values of this parameter associated in the differential equations. Okay, and when the system, when we include more components into our system, then you have more parameters to estimate or to measure, right? that sometimes make a set of sometimes make the ODE model impractical. For example, this model have more than maybe 50 equations and more than 100 parameters that we have to estimate. Okay? And if we aim to simulate more and more complicated network of the cell, for example, if we would like to simulate or to construct the whole cell model that involve a lot of components, then it would be quite difficult to use differential equations to do these tasks. Okay? So that's why the main focus of my talk today is to use another approach, which is Boolean modeling. So in this Boolean modeling, we assume that each component can have only two states, either 0 or 1. Okay, so compare this to an ODE model that each variable or each component has a continuous value, right, from 0 to certain numbers, right? But for Boolean modeling, we discretize the levels or the activities of each component into either 1, which means that the gene or the protein is active, or 0, which means that the protein is inactive. Okay, and then the interaction between components are represented by logical rules instead of the rate of change in the differential equation. We use logical rules to determine the relationships between the regulators to the target proteins. For example, if I have this small network where we have protein X and Y as the regulators, these two proteins co-regulate the activity of protein A. Then we can represent this interaction by these kind of logical rules. For example, I can say that the activity or the state of protein A at the next time step would be equal to the current activities of protein X and Y at the current time step. And then it depends on the biological knowledge about the relationships between the regulators and the target. So let's say if experiments indicate that you need both activities of protein X and Y in order to activate the activity of protein A, then we will use an AND function. Okay, so this one means that you need X and Y to be 1, to be on at the same time in order to activate the activity of protein A in the next time step, right? So we can translate this logical function into a table like this. We have all possible combination of the states of X and Y at time t, right? And then from that, we can determine the states of protein A at the next time step. So in this case, only when we have active X and active Y, right? Both X and Y are in the states of the on state that can activate the activity of protein A in the next time step. But on the other hand, if experiments indicate that either activity of X or Y is enough to promote or activate the activity of protein A, then we can use an OR function instead, right? So this one indicates that you need only one protein to be in the on state at the current time step in order to activate the activity of protein Y in the next time step. And the table would look like this. Okay? So this is the idea of translating the interaction between proteins in the network into a Boolean model. Now the function can get more complex or more complicated if we have more components involved in the regulation. For example, if you have 3 regulators X, Y, Z, then you have 9 different possible combinations of the inputs. And then from those inputs, you can calculate the output, which is the activity of protein A in the next time step. For example, in this particular example, we say that protein C is the inhibitor of protein A. So protein C inhibits the activity of protein A. And I use an AND function here. So it means that whenever C, the activity of protein C is on, it will switch off the activity of protein A in the next time step, right? And if the protein C is off, then you need at least X or Y to be 1 in order to switch on protein A in the next time step. Okay, that sounds easy, right? So next we can also connect these logical rules from one component to another component, right? Forming a network in your system. For example, this is a simple signaling cascade when you have signal A that activates protein B, and then protein B activates protein C, and in turn, protein C activates protein D. So they activate one another in a sequential order. And the logical rules are simple, right? We just have A, use the current activity of A to update the activity of B in the next time step, and B to C and C to D, and so on, right? So we keep update these logical rules repeatedly, right? And then we can see a series of change in the activity of the proteins in your network. For example, here I can monitor the activities of these proteins in my network over time step 1, 2, 3, and 4. And you see the dynamics of the activities of protein, right? Protein B is switched on first, right? At time t equal to 1. And then when B is active, it can relay the signal to the next protein, which is protein C, which is on in the next time step, and from C to D, right? And then we can plot or visualize these profiles as a picture here. On the x-axis, we have the time, right? Time step that we keep update our model. And on the y-axis is the value of the Boolean, which can be either 0 or 1. Okay, and we can see that protein B is switched on first, followed by protein C and protein D. Okay, so this is just a simple example of signaling cascades that can happen inside the cell. Now we can get more interesting pattern of the dynamics of the protein. So this little network is called a pulse generator. So we have signal A that activates protein B, and this signal A also activates protein C. And then C receives the signals from both A and B. Okay, so A activates C, but B inhibits C. So we translate this network into these logical rules. So the activity of B depends only on the activity of A, but the activity of protein C depends on A and B, like this. And B is the inhibitor, so I put not B here. And if we use this model to simulate the dynamic change in the activities of the network, you can see that protein C is switched on because first we have A, the activity of protein A, right? That's switched on protein C. But then later on, A also activates B that inhibits C. Okay, so you see that we have, after the activation of protein C, it is switched off in the next time step. Okay, so generating the pulse of the output of this network. And we can connect this pulse generator, so this is one pulse generator, to another pulse generator, and then this pulse is the second pulse generator is connected to the output protein. Then we can create patterns of a pattern of pulses of protein that are activated in a sequential order like this. C. So the output of protein C is activated first, go up and then go down, followed by protein F, and then followed by protein H, something like this. Okay. Now, inside the cell, the signaling network there are so many ways that they can interact with each other, right? One particular example that we see a lot is they have parallel signaling cascades, meaning that we have one protein that activates two pathways in parallel. For example, A activates protein B and protein E, and then each protein also activates the downstream proteins independently from the other pathway, like this. And if we translate this one into a Boolean model and then do the simulation, you would get something like this, right? B would be switched on at time step 1, and protein E is switched on at the same time, right? Because they are symmetric, right? So A activates both pathways, the pathway on the left and pathway on the right simultaneously. Okay, so you see that B and E are switched on together, followed by C and F that are switched on together, and finally protein D and protein G are switched on together. So if you visualize this profile, you see that these two proteins are on exactly at the same time, right? Because that's the way we update our Boolean model. Okay, but then we would like to get something more realistic, so we can introduce another type of updating into our Boolean model. So previously we update everything synchronously, meaning that we update all the variables in our network all at once, right? Everyone gets updated according to their according to its logical loop, right? But we can also do asynchronous updating, meaning that we choose only one variable, one component at a time, and then update that component per each time step. Okay, so we randomly select one component, update that component, and then move to the next time step, randomly select another component, and then update that component, and keep doing this over and over. Okay, what we can get here is that now your simulation becomes stochastic, right? Because there's the randomness in the selection of the components to get updated at each time step. And when you do that, I think one nice thing about this is that even you begin with the same model, right? You have the same topology of the network. You have the same logical rules for each of the component. But every time that you do the simulation, you will get different results. For example, if I simulate this network, the first time I would get something like this. For example, protein G. So we focus on the outputs of this pathway, right? D is the output of the left pathway, and protein G is the output of the right pathway. Okay, and you can compare with the previous one, right? So let me back up a little bit. with Boolean modeling with synchronous updating, all the variables get updated simultaneously. You will always get the same activation pattern of D and G, right? But if you use asynchronous updating, then the activation of protein D and G can be at different times, right? And you see that for the first simulation, protein G is activated first, followed by protein D. If I repeat this simulation again, maybe protein D get activated first, followed by protein G, right? So I can do this maybe 100 times, and each time that I do the simulation represent the behavior of one cell. Okay, so we can think about doing this simulation as the simulation of individual cells. So if you want 100 cells, you do 100 simulation repeats. And then you can align all the simulation from, let's say, 100 cells align them on the same time steps. And then we can average the Boolean values of all the cells for each time step. And then we create the profiles at the population levels of the cell. Okay, so here, for example, if I calculate the average Boolean values of protein D at this time step, right? I just use the value of each of 100 cells, average them, and then that would give me about 0.8 here. Okay, so and this is nice, right? Because we started with Boolean modeling, which only give you a discrete values of each component. But if you combine this idea with asynchronous updating, do the simulation repeats many times and average them, then you get something more continuous, right? They look like more results that you can get from a system of differential equation, for example. Okay, so you see that you see the dynamics of the proteins that stochastically accumulate from 0 to 1 over time. Okay, and here we refer to this value as the protein activity rather than the Boolean state of the protein. Let me give you one more example that might be more interesting. So the same path, the same network, but I add more another mutual inhibition relationship between the output protein D and G. Okay, so when we do the simulation of this network, we see that if protein D is activated first, it will inhibit the activation of protein G, right? So if D switch on first, then G will never get activated. But if you do the simulation again and protein G is activated first, then that will inhibit protein D, right? Because we have this mutual inhibition relationship between these two proteins. Okay, so you can simulate, again, you can simulate many cells, 100 or 1,000 cells, and each cell will have only one pathway that is activated by either D or G. Okay, and then you can average the behavior of the activities of this protein over many cells to get the protein activity at the population level. So here you see that the activity of proteins D and G go up to only 0.5, right? Because at this, when you look at each cell at this time step, you will see that half of the population have active protein D, and the other have inactive protein D. So that's 0.5. Okay. Okay. So let me say this again about how asynchronous updating works. Okay. In asynchronous updating, you select one component randomly at each time step, and then update that component based on its logical rule that you describe based on biological knowledge about your network. And then you repeat this process over and over until your trajectory, until your activity profiles of the proteins reach a stable steady state, meaning that they don't change the activity as the time increases. Okay, and then you can stop your simulation. Okay, so based on this idea of modeling approach, I apply this modeling approach to two projects. The first project is using Boolean modeling to understand breast cancer signaling pathway. And another project, I use Boolean modeling approach to study the cell cycle of yeast. Okay, so let me show you the first project. The first project is about cancer, and I'm interested in this one because statistically 1 out of 6 deaths is because of cancer, and over 90% of cancer deaths is because of drug resistance. So basically, drug resistance means that the cancer patient receive a medication and the medication failed to kill the cancer cells. Right? Maybe initially the patients may respond to the drug. The cancer cells seem to slow down, seem to get smaller, but then eventually it becomes resistant to the drug and then grow again. Okay, so that's the drug resistance. And we would like to understand how drug resistance happens in cancer cells. So for example, I show you this picture again. This is the signaling pathway that monitor the growth factor, which is the molecules that is the signals in the environment of the cells. And these signals actually promote the growth and proliferation of the cells. Okay? And in cancer cells, the problem is that there is something not normal in the signaling network, such that the cellular functions such as cell growth and proliferation is increased a lot compared to normal cells. So cancer cells, they grow uncontrollably, right? So normal cells, they grow and die, right? But cancer cells, they just keep growing and growing, right? So that's the problem. And actually, we have a handful of drugs that we can use to inhibit specific proteins in the signaling pathway, right? But because this signaling pathway promotes cell growth, right? And we don't want cancer cell to grow too much, right? So we have drugs that inhibit certain proteins, and when these proteins are inactivated, they cannot, the cancer cells, the signal cannot activate the proliferation of the cells. But the problem is that you see that, as I show in one of my examples, that usually in signaling pathways, there are redundant pathways. For example, here we have this protein that activates the left pathway, the Laomettachit pathway, and this pathway promote the proliferation of the cell. Right, so cells grow and divide, increasing the number of cancer cells. The same molecule, the same protein also activates another pathway, PI3K pathway, and this pathway also promote proliferation of the cell independently of the first pathway. So there are two pathways that work that promote proliferation, and usually when we treat cancer cell, we give only one drug. Right? For example, we can give this drug to inhibit the activity of this MEK protein. So you kill off this protein, and for sure, when you don't have the activity of this protein, you reduce the proliferation of the cancer cell. But then the cancer cells can use the other pathway, right? The redundant pathway, to compensate for the inhibition of the left pathway, and cancer cell can grow again, right? Using alternative pathway. So the idea, a recent idea of curing cancer cell is that we don't use only one. We don't use single drugs, but we will combine different drugs that inhibit different pathways simultaneously. For example, we can inhibit MEK and also use another drug to inhibit PI3K. So by combining two drugs together, it's more effective in curing cancer cells. Okay, so that is called drug combination. So the definition is that we administer two or more medications simultaneously, and this will achieve better therapeutic effect in the treatment compared to when we apply individual drugs alone. Okay, so I give you one example. This is from an experiment. The x-axis is the day after the tumor is treated with drugs, and on the y-axis is the size of the tumor. So the topmost line is the control. So this is the tumor that is not treated by anything, right? And this tumor keeps growing bigger and bigger over time. Now, if you treat these cancer cells with this drug, with this dose, it can slow down the growth of the tumor cell, right? Which is shown in the red line. If you use another drug, laloxifene, with this dose, it can also slow down the growth of the tumor cells, as you can see in the blue line, right? But if you combine these two drugs together, which is the purple line, then you see that it's more effective than using individual drugs alone, right? You combine them, and the cell, the tumor mass actually is smaller, getting smaller and smaller over time. Okay, so we say that these two drugs have the synergistic effects in curing cancer cells. Okay, so the first project, the idea is that we would like to understand how some drugs pairs, right? So we combine two drugs together, right? But not all the combination that will give you the synergistic effect, right? So most of the time when you combine two drugs, they are not more effective than using individual drugs alone, right? Only some pairs, some combination that get synergistic effects, right? So they are far better, far more effective than the single drugs. But also sometimes when you combine two drugs, they perform even worse than individual drugs, right? So we call that antagonistic effects. Okay, so this project tries to understand how breast cancer cells respond to different types of drug combinations, right? So we would like to understand why some drugs have synergistic effects on breast cancer cell, why some drugs have antagonistic effects on breast cancer cells. Okay, so this project was done by my PhD student, Mr. Kittisak Thauma. So what he did is that he constructed signaling network of breast cancer proliferation pathway, and he collected the data, the interaction of the. So these are proteins, right? The green boxes. These are proteins inside the cancer cells, and the arrows, the black arrows are the activation, regulation, and the red arrows are the inhibition. Okay, so to come up with this interaction, Kittisak, my PhD student, he used information from different databases and also evidence from literature, right? Combine them. So he has to carefully and painfully combine all the information on the public databases to get this picture, which has 117 proteins. And then after that, he translated these interactions of these proteins into 117 logical loops. So we have 117 lines, 117 loop, right? Each loop describes the regulation of one component in our network. Okay. And then after that, we do the simulation, right? Just update. So use these logical loops to update the state of the protein over time, right? Just repeat using asynchronous updating, right? So it's a stochastic simulation. Update this model over and over and over until it reaches some steady states. Okay? And so let me show you some results, right? So the network is very big, right? 117 network, and we update all of them, right, using the asynchronous updating. But for visualization purpose, I just take a tiny part of our network to show you in this slide. Okay, so it's better to understand what is happening inside our network. Okay, so here I take only 6 components from our network. And in this tiny network, we have IRS1 that activates 2 pathways. On one hand, it activates the JAK2 STAT5 pathway. And this pathway is known to promote the proliferation of the cell. On the other hand, IRS1 also activates PI3K and downstream pathway, this one. And this pathway has a negative feedback loop to inhibit the IRS1. So you see, if you start from this one, IRS1 activates PI3K and then activates AKT, mTORC1, and S6K1 in sequential order, right? But at the end, S6K1 inhibits the IRS1 protein, forming a negative feedback loop. Okay, and we can do the simulation. So again, we do the updating for 117 proteins. Okay, but here I just show you only 6 proteins. But this is the result from the simulation of the whole network. Okay, so we can see again we do asynchronous updating, right? So one simulation represents the behavior in one cell. So we do, if I remember correctly, we do 500 cells and then average the Boolean values from 500 cells to get this picture. So this is the protein the activities of each protein in our network. Okay, so it go up and down like this. And the idea is that we use our model to see how the network, the proteins in the network responds to the perturbation by the drugs. So at this time step here, we apply the first drug, BEC235. So this drug is an PI3K inhibitor. And we model this inhibition by just setting the activity of PI3K to be 0. Okay, so when we apply the drug at this time, after that, the activity of PI3K remains 0 until the end of the simulation. Okay, so this represents the inhibition effects by this drug. And after that, we see how the inhibition of the activity of this protein affects the other activities of the rest of the protein in the network. And what we see is that once PI3K is downregulated by the drug, then IRS1 and JAK2 activities go up, right? So previously it go up and down around 0.05 to 0.75, right? But after we apply the first drug, it goes up to 100%, right? And that is understandable, right, from our network here, because if you turn off this protein, this activity of this protein is 0, then this one goes down, right? AKT is switch off, mTORC1 is switch off, S6K1 is switch off. But that is the inhibitor of IRS1. So it means that IRS1 will be turned on, right, after you apply this perturbation. And that is seen, clearly seen by the simulation here. Okay. And when IRS1 is on, it cannot activate PI3K, right? Because we have the drug that keep it inactive. But it can activate an alternative pathway, which is JAK2 and STAT5 over here. So you see that the activity of JAK2 is also increased after we apply the drug. And that's how we explain the resistant mechanisms of breast cancer cells to this drug. Right? So when you apply this drug, you seem to slow down the proliferation by this pathway. But then we also activate another pathway that also promote the proliferation of the cells. Okay? So next, it's now quite rational, right? That you should inhibit both pathways together, right? So instead of applying only the PI3K inhibitor, we know that if we do that, then the JAK2 STAT5 will be activated. So we can also use another drug, which is the JAK2 inhibitor, to kill this pathway as well. Okay, and that is actually the simulation that reproduce the observation from the experiments. So in this experiment, they apply different drugs to breast cancer cell. So the y-axis is the size or the volume of the breast cancer. The black one is the control, right? When the cancer cells are untreated, so they grow over time, right? After 19 days, they grow very big. The green line is when you apply the JAK2 inhibitor alone, okay? And it doesn't help at all, right? You see the green line is actually quite similar to the black line. And the reason is that because in this system, IRS1 is downregulated by S6K1, right? So actually this pathway is not quite active in the first place, right? They are not active. So if you apply the inhibitor of this pathway, it doesn't help, okay? But then if you apply one drug, which is the PI3K inhibitor, the red one here, then it slows down the growth of the tumor, right? So you compare the black line and the red line, right? You slow down the growth of the cancer cells. But again, that would also reactivate the JAK2 STAT5 pathway, right? So when you combine both PI3K and JAK2 inhibitor together, that's the blue line, right? Then you get better effective effects of the drug, right? So this one has the smallest tumor size compared to the other treatments. Okay. So let me show you another example from the network. So in this case, we have EGFR, which is another important protein in breast cancer signaling pathway. EGFR activates the last MEK pathway that eventually promote the proliferation. EGFR also activates the PI3K AKT pathway that also promote the proliferation. Okay, so if we apply a single drug that inhibits MEK, okay, and from this interaction, we see that if you turn off the activity of MEK, you will turn on the activity of EGFR. And you can see that from our simulation here. So when at this time step, we apply the drug like CI, the drug name is CI 1040, right? And we switch off the activity of MEK, and we see that the activity of EGFR goes up. Okay, it just go up and down a little bit because it's a stochastic simulation. Right? What we see is the average dynamics from many cells. Okay, so the idea is that we can, and if EGFR is on, then it will activate this pathway, and the cell will remain proliferative, right? So the idea is to combine the inhibitors of MEK and also the inhibitor of PI3K simultaneously to kill both pathways. Okay, and again, this is the simulation that we produce, an observation from an experiment here. Again, if you apply a single drug, this is the MEK inhibitor, right? So you kill the cancer cells to this level. If you apply another drug, the PI3K inhibitor, you kill the cancer cell to this level. But if you combine them together, which is the lowermost line here, you combine two drugs together, it is far more effective than when you use single drugs alone, right? And you can also see that the dose that we use here, the dose that they use for the drug combination, the first drug, the dose is 2.5. The second drug, the dose is 2.5. So this dose combination, which is very small, right, has the more effective effects than the dose here. Right? Here we use 10, right? So each drug, the dose of each drug when used alone is 10, and 10, right? Here we only use 2.5, but you get better therapeutic effects to killing the cancer cells. Okay, so more effective and lower dose that we use. Okay, so we use that. So after we get this model and we think that it captures some experimental results that has been reported in the literature. Now we use this model to simulate over 100 drug combinations. So we gather a list of 150 drug combinations from a database. Okay, and then we simulate the effects of this drug on our network. One highlight of our study is that we focus on 42 synergistic drug pairs. So these 42 drug combinations have been shown to be effective in curing breast cancer cells. So we simulate the responses of the protein in the network to the perturbation by these 42 drug combinations. And we classify the responses into 5 patterns. Okay, so from 42 drug combinations, they can be classified into 5 patterns of protein activity responses. Okay, the first pattern is that these 2 proteins are highly upregulated. So the activity of these 2 proteins are very high in response to some of the combination of drugs that we use. Okay? And these 2 proteins are the ones that promote cell death. Okay? So we know that now we understand more about the underlying mechanisms of the drug combination. We can predict that these drug combinations work very well because they promote cell death. Okay? And also other patterns such as the downregulation of another protein, BCL2, which the downregulation of this protein also promote an alternative cell death pathway. The third pattern is the downregulation of mTORC1, which inhibits protein synthesis and metabolism. Some drug combinations reduce the activity of ESR1, which inhibits the response of the breast cancer to the hormone. And the last pattern is when drug combinations reduce the activity of cyclin D, which inhibit the cell cycle process. Okay, so we propose that there are 5 patterns of mechanisms that we can use the combination of drugs to slow down the growth of breast cancer cells. Okay. Another project, we apply the same idea using Boolean modeling to the budding yeast cell cycle. Budding yeast is a single cell fungus that is used in baking, right, in bread, right, making bread, and also brewing, right, making alcohol beverage. We are interested in budding yeast because the cell cycle regulation in budding yeast is very similar to what we have in humans. So budding yeast is a model that we can use if we understand the regulation of the cell cycle in budding yeast. Maybe we can understand more about what is happening in human cells. Okay, and we started by having only 7 components in our network. So this is the first version of our model, having only 7 components, and they are regulated. They regulate one another in this diagram. So all these regulations are derived from biological knowledge that we get from literature reviews. Okay? Let me tell you about some proteins, some important proteins. For example, CRIN2, this protein promotes bud formation. Okay, bud formation. So if you look at this picture, you see that the yeast cell, when it enters into the cell cycle, it will create a small bud like this one. So this bud eventually grows bigger and bigger and become another cell. Right? So this is the way that one cell create another cell and become 2 cells. Okay, so this is the cell cycle process. Okay, and the formation of this bud is regulated by CRIN2. It means that if CRIN2, if the activity of CRIN2 is active, it corresponds to the formation of the bud of budding yeast cell cycle. Another component, CRIP5, promote DNA synthesis. So because budding yeast undergoes the cell cycle process, they create another cell, right? So they have to replicate another copy of DNA, and then these 2 copies of DNA will be distributed, one to the old cell and another to the new cell. And CRIP2 is another protein that promotes mitosis. So mitosis is the distribution of DNA to the two new cells. Okay. And again, we translate this diagram into logical rules like this. So we have 7 equations, seven logical rules, and then we do asynchronous updating. And because we have 7 components, so we have 128 possible states in our system, right? So this is the one of the 128 possible states of our system, which is represented by a number of 7 digits. For example, state 1 followed by 6 zeros is when we have one of the components, CDH1 is on, that's the first digit. And the other components are inactive, right? They are all 0 here. They are all 0 here. Okay, so we can simulate our model and track the trajectories of the change of the state. Right? For example, here we have, let me zoom in. Here we have 1, 1, 0, 0, 0, 0, 0, right? And if I update this state using that 7 logical rules, then I will get the next stage, which is 1, 1, 1, 0, 0, 0, like this. And I keep doing that, and then connect this transition into this network. Okay, and what is interesting is that in this system we have this about 11 states that correspond to the normal physiology of the budding yeast cell cycle. Right? We can correspond this state into the physiology of the cell. For example, this one here, this is CRIN2. This is when CRIN2 is on, and that means that the cell has the bud formation. Right? When CRIP5 is on, which is the fourth digit of this number, CRIP5 is on, and that represents the synthesis of DNA. Okay, so you see that the state of the system update in this order, right? And that represents different phases of the cell cycle. And what is more interesting is that these 11 states are the global attractor of the network, right? Because we have 128 possible states. If you start from any state in this picture, it will eventually converge to this global attractor, which represents the normal physiology of the cell. Right? So you can start from some state that is not normal physiology, but the Boolean, but the regulation of these proteins will update one another and correct the state into the normal physiological state, which is represented by this attractor in this picture. We also update the second version of this model with 22 components, which is far more complicated. We also upgrade how we calculate the time, right? So previously, the time in the Boolean model is just discrete, right? Just time t equal to 1, 2, 3, 4, 5, and so on. They are discrete, and it doesn't have any biological meaning, right? So we try to somehow calculate the update of the time in our system so that it can have biological meaning and can be compared to experiment. So in this case, we know that for example, if we consider the production of protein A, okay, so protein A is produced from 0 to 1 molecule, from 1 to 2 molecule, to 3 to 4 to 5, right? And we know from the principle of chemical reactions that the intervals between chemical reactions are exponentially distributed. So the time interval, which is the the length that is pointed by these red arrows, they are exponentially distributed. Okay. Then we assume that each Boolean value change is a series of k elementary steps that are independent and identically distributed random variables from the exponential distribution. Okay, then we can assume that the delta t here, so this is the interval between the Boolean state from off to on, will follow the gamma distribution with the shape parameter equal to k and the rate parameter equal to lambda. Okay, so now we can update the time using this gamma distribution. So now time is also stochastic and continuous, right? So I compare the simulation from my previous project from breast cancer modeling. In that project, the time step is just the time step that we update from 1 to 2, 3, 4, 5, 6, and so on. Right? It doesn't have any meaning. But for this budding yeast cell cycle model, now the update of time is also has a unit of minutes. Okay, and by doing this, it enables us to calculate certain quantitative properties from our model. For example, we have time of the time to complete one cell cycle process. Okay, so we can measure that from our simulation results and compare that directly to the experiments. So in this picture, the blue bar is the simulated result, and the orange bar is the experimental observation. Okay, so which is very nice. We can we started from very simple Boolean model, right? That is discrete in terms of the values of the components and also the time, right? But now we can use these kind of techniques to convert the values of the components to be continuous, and also time in our model is also continuous and stochastic. Okay, for the conclusion, I try to convince you that Boolean modeling is a very powerful tool to study dynamics of complex biological networks. And in the first project, we applied this approach to study breast cancer signaling network, right? And I think the one thing that I would like to emphasize is how we convert the discrete values of each component into the continuous protein activities, right? Using asynchronous updating algorithm, right? And these quantitative values of protein activities allow us to identify key proteins that are significantly perturbed by the treatment, by the application of the drugs. In the second project, we also implement a method so that time in the model is also continuous and stochastic, and that allows us to calculate some quantitative properties from the model that can be compared directly to experimental data. Okay, so I would like to thank many people who contribute a lot to this project. Mr. Kittisak Thauma, my PhD student, he is the main contributor to both projects. Dr. Marasi Leungkit from KMUTT and Dr. Moridi Leraksa from Nanotech. These are my collaborators in the Boolean modeling project, in the breast cancer modeling project. Professor John Tyson, my former advisor at Virginia Tech, and also Professor Paweł Kajkiewski, also from Virginia Tech, for the second project in the budding yeast cell cycle. I also would like to thank KMUTT and the Pechpachomkao PhD Research Scholarship from KMUTT and NSTDA, and also a funding from Thailand Science Research and Innovation Basic Research Fund. Also, thank you everyone for your listening. Sorry that I took so long. So if you have any questions, please feel free.
All right. Thank you very much for a very, very nice talk. I think it's about time for questions.
Thank you for a very interesting talk. So I have a few questions. So there are these differential equation model, this synchronous updating Boolean network model. So should I think of them as completely different models, or if one is approximating another? Like, do you expect if these two models apply to the same phenomena, one would be more accurate than the other, or it depends on what you're trying to do?
Yeah, okay. Yeah, very, very good question. So I think so if you want to describe your system accurately, then the differential equation model would do a better job, right? Because it capture the kinetics parameter and dynamics of the proteins, which is more accurate for sure than the Boolean modeling. So it depends on the level of accuracy that you want from your system. Usually when the system is very big, like for example more than 50 components in the network, it's quite difficult to start your project by developing an ODE model. So we usually start from using Boolean model first to get some understanding of the system. And later on, if you would like to get more accurate detail of your system, you can extend your Boolean modeling to have some. So there are also another type of modeling approach, which is the hybrid modeling approach, when you combine both Boolean variables with differential equations. Okay, so some components can be described by differential equations, and some components can be described by Boolean networks, right? And then you combine them together. Okay, so that's the hybrid one. And later on, if you want to add more and more detail and get more accurate prediction, maybe you can translate everything into ODEs.
Okay, very interesting. Another curiosity is in the yeast flooding model, you talk about the attractor of it. So, like for people working in this field, usually like how do they find attractors or predict attractors? Like, are they just doing numerical simulation, or is there something that's do analysis?
Okay, For my research, we just do numerical simulation. So because it's a stochastic simulation, so we just repeat the simulation many times, like 5,000 simulations, and then just identify the attractors from that. But I also believe that there would be some kind of analytical method to do that too. That's beyond my knowledge, sorry. But I think that there are some works that do this kind of analysis as well.
Thank you.
Okay. Okay. I have questions about your Boolean modeling simulation. How do you expect that there are maybe, for example, 3 or 4 branches from, let's say, from the first activator A to B the branch that you showed us earlier. How did you know that there may be 2 or 3 and probably this one has a pulse system? How do you identify?
Okay, I cannot hear very well, but if I understand correctly, usually we start by developing a simple model first. So we try to make our model as simple as possible and see if the model can explain or capture the behavior of our biological system or not. And then after that, we would see that there are some discrepancies between our model simulation and some experimental results or experimental observations. Then we have to refine the model. Sometimes we add more interactions. Sometimes we add more nodes, more components, and see if it can address the experiments that we could not explain in the earlier model or not. That's the way how we do it. So for example, in our first version of the budding yeast model, we have only 7 components, and this is very simple. This model is very simple, but it cannot explain a lot of experiments. That's why we have the second version that we add a lot more components into the model, a lot more interaction between the nodes in the model. I'm not sure if I answer your questions.
Anyone else would like to? Well, I just have like a naive question. So, do doctors need to know your to cure the patient? So you just so it means that you just give them only your result, right? Or like or possible combination of the drugs. Is that what?
Can you repeat the question again?
What I'm asking that actually, does doctor need to know your math model?
Oh, okay. Okay. Yeah, I think right now, the doctors have a protocol, right? They have to follow the guidelines of giving the medication to the patients. So I guess our research is try to identify new therapeutic approaches. But in order for this kind of finding to get into clinical use, it has a lot of stage that we have to prove, right? We have to do it in the laboratory first, and then it has to go into the clinical trial many years before it can be used.
Okay. So, can I ask just one more question about your Boolean model? If I understand correctly, this model is just kind of step forward, right? It's kind of step forward sequencing. So it means that in the bio system, so the way that actually the node or the protein node works is only the step forward sequencing, right? There is no backward, because if I understand, actually from your Boolean model, it just kind of step forward, right? Sequence.
Actually, some proteins can. So in biological system, there are a lot of feedback loops as well. Okay, so it means that the protein on the top of the pathway can get regulated by the protein from the bottom of the pathway as well. So I think it's not just going from top to down, but it can go from down to top as well.
But what about in between? In between?
Yeah, in between as well.
Oh,
So they are quite complex interactions.
Okay, right. But one of the advantages of your the way actually you model thing with the Boolean technique is that there are no parameters in your model.
Right, yeah.
Yeah, that is I think that is the
yes. I forgot to mention that, and thank you for pointing that out. Yeah, there's a parameter free.
Yeah, because if you stick with the a set of differential equations, you need to deal with a lot of parameters. But I don't see this type of parameters appear in your Boolean in the Boolean model. That would be the nice thing, easy to work with.
Yes, exactly.
All right. And I believe that actually even this kind of research still like in the baby step, but I think I see kind of great potential. Actually, it would help a lot of people in the future, right? Because we can if we understand the underlying mechanism to prevent somehow the cell cancer to grow exponentially, so somehow that would be good, right? And so another question is that, so what about some other kind of cancer? So because you're now focusing on only the breast cancer, if you start with some other kind of the cancer, that means that you need to repeat the whole thing again, right?
Yeah, I think that is a challenge in cancer research because different types of cancer, they are quite different. Even in the signaling pathways, because they have this cancer, they have mutations, right? Mutations means that some of the protein might function too much, and some of the protein do not function at all. So if I switch from breast cancer to do let's say lung cancer, then I have to update all of those information into the model. Right?
All right. It's still a lot of work needs to be done.
Yes, but I believe that the workflow, I mean the methods that we develop should be the same.
All right, I see. But the inside internal structure might be different, right?
Right. Yes. And even in the same cancer with different subtypes, they are quite different from each other as well.
I see. Interesting. This is this is why it's very complex, right? This is somehow it's a complex system. I think this is all for all of us. Is there any online question? Maybe no online question. Then let's start speaking. Okay.
Thank you for having me, huh?
All right. Okay. So that's all for today. And so we're going to have the next colloquium this month again on 24. Right. Then keep tuning on that, and then see you. Bye bye for today. Thank you very much, everyone. Bye. ขอบคุณครับ